Hello everyone and welcome to the Digestive System Lab. This will also have two components like the respiratory system. There's anatomy and physiology portion. And just like the last time, I'm going to start with the physiology portion and then we'll get into the anatomy. So everything you need to know about the physiology is actually on this Canvas page. So again, similar to how I had it set up for the respiratory system. Your objectives are listed right here, and I tried to make all of that information presented in this Canvas page exactly in the same order. So you need to know the three key things about enzymes, the difference between chemical and mechanical digestion, what breaks down carbohydrates, proteins, fats, and how carbohydrates, proteins, and fats are broken down, and then what are the optimal pHs for the enzymes described below. Okay, so starting right off the bat, the three key things to know about enzymes are, they are highly specific, they function at a specific pH, and that high temperatures denature enzymes. And then just below you'll see there's explanations that go into a bit more detail. So for enzymes being highly specific, basically this means that even a small change in a molecule means that the enzyme cannot digest it. And here's a classic example of that is the difference between sucrose and sucralose. They're very, very similar. You'll notice just right here there's a bit of a difference. And then right here, there's a little bit of a difference, but that's enough of a difference in sucralose, which makes it 0 calorie because your body cannot digest it and use it. If it could, it would now have calories associated with it. Okay. And then here's enzymes function at a specific pH. So again, this is a very important concept to understand about our digestive enzymes. We will have to have multiple digestive enzymes that break down similar substrates to get them fully broken down, because as food moves from our mouth, which has a pH of like roughly 7, to your stomach, which has a rough pH of about 2, back to your intestines, where again the pH is roughly a 7. When you're going from an environment that's a pH of 7 to 2 back to 7, those are huge changes. So an enzyme that works in the mouth and in the esophagus will stop working in the stomach. Something that worked in the stomach will stop working when you get to the intestines, because again, you're going from a pH of 2 to 7. So that means that in those areas, you're going to either have to release new enzymes, or simply digestion of whatever that substrate is cannot occur in that place. Okay, and then just also to be aware, high temperatures will denature enzymes. That means that their structure is permanently altered, and once it's altered, it will no longer work. Okay, freezing does not work the same way. You can freeze enzymes all day long down to minus 80 degrees Fahrenheit, and once they thaw, they will work just fine. However, heating them up will destroy them. Okay, now as far as specific enzymes go, the ones that we want you to be aware of are these for carbohydrates. So amylase is the main one. It's produced, there's a version of it in saliva. There's a slightly different version of it being produced by the pancreas. And you can see here, since it's found in the mouth and in the pancreas, and those areas are both associated with a pH of 7, that's where it will function. So the amylase that is produced in the saliva, once it gets to the stomach, it stops working. So that means that's why you have the pancreas amylase or pancreatic amylase that will release into the duodenum, the first part of the small intestine, because again, that salivary amylase has stopped working in the stomach. So now you need a new one to start back up in the small intestine. For protein, there's a lot of different enzymes that will break down protein. You'll see more of that information in lecture material. We're just going to focus in on pepsin here for lab. Pepsin is produced by chief cells in the stomach, and you can see this. What they're actually producing is a precursor called pepsinogen. That O-gen, it means it's coming before. It's a precursor molecule. That pepsinogen is quickly converted into pepsin by hydrochloric acid in the stomach. Again, it functions at an optimal pH of 2. This means that you will have different things that would work on proteins later on in the intestine, but we're not going to worry about that for lab. You'll hear more about that in lecture. Fat is a large molecule. It's actually a 2-step process. The first part is mechanical digestion, and what does that is going to be bile that is secreted by the gallbladder and the liver. Mechanical digestion is a physical digestion, so meaning that you're simply chopping up the molecules into smaller pieces. It's no different than cutting something up with a knife. That would be a mechanical digestion, or in your body, like chewing something with your teeth. That is mechanical digestion. You're not chemically altering its properties or nature or anything like that. You're just chopping into smaller pieces, and bile does that. Okay. So, and that's an important first step, because you want to take these very large molecules, chop them into smaller, more bite-sized pieces, for lack of a better way to say that, for the enzymes to then break them down, okay? Because if it's too large, it's hard for the enzyme to break it down efficiently. I always liken it to trying to eat a watermelon before cutting it up. You could do it, it's just going to take a lot of work, and it'll probably be really messy and slow. Anyway, so the first step is mechanical, and that's done by bile, right? And again, we're physically breaking these fat molecules into smaller droplets, but they're into smaller pieces. The next step involves lipase, so that is an enzyme, and that because it's an enzyme, it's going to be a chemical reaction. That lipase is also being secreted by the pancreas. Again, so it's a chemical breakdown, and we're breaking down those smaller fatty droplets into glycerol and fatty acid. Okay. So again, that the chemical breakdown takes that fat molecule and breaks it into glycerol fatty acid. It actually changes what it is. Mechanical digestion is still a fat molecule, it's just a smaller piece of one. Okay. And here you see, because the liver, gallbladder, and pancreas release their contents into duodenum, the optimal pH is going to be a 7. Okay. Okay, so that's the physiology portion, and because this is a little bit of a longer explanation, what I'm going to do is record a separate little intro for the anatomy part.